Yeah, hi friends. So this video will discuss the limb development. So how the limb formation takes place. So limbs are present on the lateral side of the body, and in case of fish, pectoral and pelvic fin are present, which correspond to the anterior and posterior limb. In case of birds, wings are present. So it is divided into stellopod, zygopod, and the autopod. So usually upper limbs begin development first, when the limb often lagging 2 to 3 days behind, and at the fourth week of the gestation period, and it's completed about the eighth week. The area where the limb formation takes place. So first the limb fields are formed, then the limb bud are formed. So limb bud are the precursor structure of the limbs, and from limb formation results a series of reciprocal tissues and interaction between the mesenchyme of the lateral plate mesoderm and the overlying ectodermally derived epithelial cells. So the lateral plate mesoderm are the region of the mesodermal cells, which is of four types. So limb development begins with mesenchyme cells proliferate from lateral plate mesoderm and from somites. So, limb is divided into three dimensions, that is proximal and distal end, anterior and the posterior end, and dorsal and the ventral end. So, in the limb board, there is a apical part of the limb board known as the apical ectodermal ridge, which consists of central core of undifferentiated mesenchymal tip with a larger of ectoderm called as the AER. As growth proceeds, the proximal mesenchyme loses signal from the AER and begins to differentiate into the constituent tissues of the limb. So AER reciprocatively secretes FGF8 and FGF4, and which is maintained by the FGF10, and FGF10 expression regulated by the WNT genes. And in case of the forelimb, the WNT8C is present, and the hand limb WNT2B is present. So the position of the forelimb and hind limb are specified by the TBX gene, that is transcription factor genes, which is TBX5 and TBX4 for the forelimb and the hind limb respectively. So zone of polarizing activity is guided by the SHH gene, that is sonic hedgehog gene protein, act as a morphogene and determine the anterior posterior axis. So there is a Hox gene, which is for limb. There is a Hox9 and Hox10, and for the hind limb, Hox10 is present and Hox9 is absent. So Hox is further divided into four clusters, that is Hox A, Hox9, Hox-C, and Hox-T. So limb bird will develop specific Hox gene, which is further regulated by the retinoic acid. So ZP and the AR signaling centers are linked to each other through the mutual cross regulation, and there is so much genes expression like SSH gene, FGF, Ox gene, TVOX, and WN families all plays a vital role in the cell signaling and differentiation to regulate and promote successful limb formation. So many studies are focused that enhancer and SHH gene, that is sonic hedgehog gene, is required for the proper limb formation in several vertebrate species. So figures specify the position of the AER, the apical ectodermal ridge, the zone of proliferation, and in this posterior base there is a zone of polarizing activity, that PA, and this is a mesenchymal cell differentiator, which is signaled by the apical ectodermal ridge. So this is the figure of the mechanism of the limb development and the genes involved in this development of the limb. So axis formation. So three types of axis, that is proximal, distal axis, anterior posterior axis, and dorsoventral axis. So proximal distal axis is generation of proximal distal axis of developing limbs are determined by the ectodermal apical ectodermal ridge, that is AER, and it is caused by the FGF10, and this FGF10 is secreted by the lateral plate mesoderm. So anterior posterior axis is defined by the ZPA, that is zone of polarizing activity, and is stabilized by the interaction of FGF8 from the AR and mesenchyme made competent to express SH gene by its expression of particular Hox gene. Dorsoventral, it is formed by the expression of WNT7A in the dorsal portion of the limb ectoderm. So FGF4 and SHH reciprocally maintain each other expression. Digit formation. Digit formation is guided by the bone morphogenetic protein, and it is maybe the BMP2, BMP4, and BMP7. And as elongation continues, the mesenchyme condenses into plates, forming a cartilaginous model of the future digital bones. So AER then break up and maintain only over the tips of the future digit. The interdigital space are progressively found by the cellular apoptosis. So mutation or other factors resulting the abnormalities of the limbs, that is an amelia and the meromelia, and for digit abnormalities, that is syndactyly fusion of the digits, and the polydactyly increase in number of the digits. So congenital limbs and digit effects occur in between 1 and 500 and 1 in 1000 live births. So, as a conclusion, the vertebrate limbs are appendages that form crucial in being involved in locomotion, feeding, communication, and other purpose. The limbs are present at the lateral side of the body. So, all about the limb development. Thank you. Keep watching.